Professor Maxfield is mainly interested in atherosclerosis and cholesterol trafficking, in vesicular trafficking of cholesterol and non-vesicular trafficking. He's working on that field since 1998. 88. So please, Professor Maxfield.
Okay. Well, merci beaucoup for the invitation. And I'm afraid that's the last French you're going to hear from me in the Société Francophone. Anyway, it's a pleasure to be here. So this is where I work at the Weill Cornell Medical College in New York City, right next to the Rockefeller University and across the street from the Sloan Kettering Institute. And our body of water is the East River, which you can barely see there. So what I'd like to talk to you about today is an unusual pathway for cholesterol transport, I think maybe unique, that we found for the way macrophages take in cholesterol from atherosclerotic lesions. But just so everybody's on the same page, in almost all tissues, including macrophages under most conditions, the pathway for lipoprotein endocytosis and catabolism is illustrated here from the work of Brown and Goldstein. You have LDL binding to LDL receptors, gets internalized by receptor-mediated endocytosis, goes through endosomes. The receptors mostly recycle back to the cell surface. The LDL is delivered to hydrolytic organelles, late endosomes, and lysosomes, where it encounters lysosomal acid lipase that hydrolyzes the cholesterol esters and the triglycerides. The free cholesterol then has to be carried by NPC2, delivered to NPC1, inserted into the limiting membrane, where it can then be exported around the cell by both vesicular and non-vesicular transport pathways. So this is very much the conventional pathway, as I said, for virtually all uptake of lipoprotein cholesterol. But it's a little different in the case of atherosclerotic lesions. So as illustrated here, monomeric LDL can cross through the endothelial lining, but it gets acted on by various enzymes, sphingomyelinase, phospholipase, lipoprotein lipase, and others. And this causes the LDL to aggregate and additionally to become cross-linked to the extracellular matrix, as you can see here in this electron micrograph from Joy Frank a number of years ago. And what it shows is that these lipoprotein aggregates in the vessel wall are tightly cross-linked to the extracellular matrix. The collagen is conveniently labeled with a C in this micrograph, but other matrix elements are seen here. And on account of this, even though this might be a particle that a macrophage could phagocytose, it can't because of the tight connections to the extracellular matrix. Nevertheless, the cholesterol inside the lipoproteins gets delivered to the macrophages, gets esterified, and forms foam cells, which are an important aspect of the atherosclerotic lesion. So a number of years ago, we puzzled about this. How can it be that the macrophages get the cholesterol esters and hydrolyze them when they can't even phagocytose these aggregated lipoprotein particles, which, by the way, are the major form of cholesterol found in atherosclerotic lesions. I know there's oxidized LDL and so on, soluble monomeric lipoprotein particles, but the vast majority of lipoprotein cholesterol is in these aggregates. And so we decided to try to understand how do things too large to be phagocytosed get into a lysosome and get hydrolyzed. Well, it turns out it was the wrong question because they don't get into the lysosome. What happens is the lysosome comes to the particles. And so this is a cartoon model of some things I'm going to summarize some evidence for in a few minutes. But this is, and I'll show you some micrographs of this, this is an aggregate of LDL. There might be in a lesion, or we can make models of aggregated LDL in the laboratory. And so the aggregated LDL gets deeply invaginated, actually, into the macrophage. The macrophage membrane forms invaginations. It also makes tight seals. And what it can do then is it can actually pump protons. Macrophages normally have vacuolar ATPase on their plasma membrane. They can pump protons and actually maintain a slightly acidic environment outside the cell, topologically outside the cell. This is, the tight seals are maintained in part by the polymerization of actin, and there is lysosome secretion, including the enzyme lysosomal acid lipase, that gets secreted into this environment and then can digest the lipoprotein aggregates outside the cell. Now, it took us a long time to come to this idea, and I'm going to try to present to you in a few minutes why we think this is what actually happens. We've also worked out some of the signal transduction pathways leading to lysosome exocytosis and also to the control of the actin cytoskeleton. Okay, so we actually had observed this phenomenon, and other people did too. Howard Kruth and we, about 20 years ago, observed that there were invaginations of lipoproteins. Lipoproteins in these slides are going to be always red, and in this case the macrophage is green. And in this confocal image you can see what looks like a piece of the aggregate penetrating into the lysosome. But we could tell that it was actually surface connected, because after a few minutes, if we put a membrane-associated fluorescence quencher in, all of that fluorescence, but that wouldn't cross membrane barriers, all that fluorescence gets quenched, indicating that all of this stuff, it was actually topologically outside the cell. And we can see other images of this by confocal imaging. I'll show you better images in a moment. But we can also show that there is, in fact, hydrolysis of the cholesterol esters outside the cell. If we vigorously wash when aggregates of LDL are on top of macrophages, as illustrated in these micrographs here, we can see the aggregated LDL, again, in red, and you can't see it here because it's all covered up by the phalloidin stain, indicating filamentous actin in that same region in these control macrophage cells. But we can also use this diphilippin, which binds to unesterified cholesterol, and we can see that there's a large amount of unesterified cholesterol associated still with this aggregated LDL. And we can measure this biochemically. We can show that there is, in fact, the release of free cholesterol outside the cell. And we can block this process using inhibitors, even specific inhibitors of lysosomal acid lipase. And you see now there's much less philippin labeling and less stimulation of the actin polymerization. So somehow lysosomal acid lipase, which requires a low pH, and of course its presence in the space, is hydrolyzing these extracellular lipoprotein particles. And there is one other well-known example in biology of this, and this is osteoclasts, which are a classic example of extracellular hydrolysis by lysosomal enzymes in terms of bone repair and demineralization, so osteoblasts can come by and repair the bone. Now we've seen this also in macrophages, and I'm not going to show you, but we've seen it also in dendritic cells and in microglia. Oops. Okay, so one way we can demonstrate this process is we can pre-endocytose biotin fluorescein dextran into the lysosomes of the macrophages. This exits very, very slowly in an unstimulated macrophage. We can then put the macrophages in the presence of streptavidin-labeled aggregated LDL, which will capture any biotin fluorescein dextran if it gets exocytosed. And then we wash away the uncaptured biotin fluorescein dextran. And what you can see here is that after about 90 minutes, a large amount of biotin fluorescein dextran that was in lysosomes inside the cells has now been deposited onto the aggregated LDL, shown here. And we can block this in various ways. A simple way is to block the microtubules, which are necessary to transport the lysosomes to the plasma membrane, and then we get none of this secretion. We can also demonstrate that there are acidic compartments by putting pH-sensitive dyes into the aggregated LDL and seeing what happens when macrophages come into contact with them. And what happens is that we see acidification to a varying degree when the lipoproteins are in contact with the aggregated LDL. This can be seen pretty clearly in this image, which is part of a movie. And you can see here, this is reporting pH 7.4, where it's not touching the cell. Here, it's heterogeneous, but in places it's down to around pH 6. And the other thing you see from this image is that it's cell autonomous. This cell is reacting, this cell not so much, and this cell, can't be bothered. So this actually makes it a challenge to study this process. I'm going to show you a video, if I can. We can play this video. Yeah, so every jump is 10 seconds. And what you can see is that this is a very dynamic process. And over time, this cell, which is actively engaged, actually does finally bite off pieces of the lipoprotein and deliver it into real lysosomes where the hydrolysis continues. And we haven't stained this, but I think that these clear areas are some lipid droplets that are already forming after 20 or 30 minutes or so of this interaction. So we wanted to see this in higher resolution, so we looked in electron microscopy. First, in a tomography experiment, and this is with colloidal gold labeled LDL, and you might be able to see the little black dots of colloidal gold on the LDL particles. And this indicates what a complex structure is formed when the macrophages interact with the aggregated LDL. And you see some things like this that almost look like they're in an organelle, but if you look carefully, there's a connection out to the outer space there. We can only see about 200 nanometers at a time using EM tomography. So we switched to focused ion beam scanning electron microscopy, again with colloidal gold LDL, which you can see here. And if we can play the video again. Here we go. So we're going to go through the cell. We etched away 40 nanometers at a time. We went through about 500 images, or 1,000. And then we filled in, everywhere we saw colloidal gold, we filled it in with red here. And the surface we filled in with gray. So you can see there's this one aggregate of LDL that is penetrating quite deep into the cell, almost to the nucleus. And it's a complex structure. These things that are now in green are inside the cell, but still topologically outside the cell because they're connected to the outer parts. And you can see that there are little knobs on this, and I'm going to come to that in a moment. So this is the macrophage forms a complex structure which we think is important for it to be able to create the tight seals. And this is further illustrated in this ARIScan confocal image, where, again, the plasma membrane is gray, the lipoproteins are red, and now we've got F-actin labeled with phalloidin. And you can see, actually, in some of these knob-like structures, that there is actually a ring of actin that's sealing that off, making it so even protons exit slowly, and you can acidify this region, and you can have lysosomal hydrolases in there, and you can actually digest these pieces of this aggregate of lipoprotein. It's all one aggregate, and topologically, it's all outside the cell. even though it's penetrating several microns in. So we think that this is something we call a lysosomal synapse. And the idea is that the macrophages and all our dendritic cells interact with the aggregated LDL. They form these invaginations. And the result of this is that as they seal with the assistance of the F actin, you get an acidified region from the vacuolar ATPase. You get secretion of lysosomal enzymes, including lysosomal acid lipase. They digest away these lipoproteins. And importantly, the cholesterol gets delivered directly to the plasma membrane. So there's no intervention of NPC1, NPC2, anything else. The cholesterol goes right into the plasma membrane. And a consequence of that is that it sets off various signal transduction processes, including increasing the activity of CDC42, which leads to the actin polymerization, decrease of Rho, which is actually from the ceramide that's released from the lipoprotein particles. And so you get signal transduction happening at the plasma membrane as a consequence of this mechanism of uptake of cholesterol from lipoproteins. So everything I've shown you so far has been from cell culture studies. What about in vivo? So this is a mouse, an ApoE mouse, ApoE knockout mouse on a high-fat diet. And so here we stain for ApoB, to see the lipoproteins. We stained in red now for philippin to see the unesterified cholesterol that was already released from these lipoproteins. And we see that the philippin and the lipoprotein ApoB are all in the same places in this lesion. If we look at lower magnification, just at the philippin labeling, you might be able to see about 100 little dots of philippin in here. And if we look at that at one of these, or any of these at higher magnification, this is what we see. Here, actually the macrophage is labeled red with an antibody that recognizes the macrophages. We're just looking at the unesterified cholesterol, the philippin, and this is a single confocal slice through this. This is a 3D projection, and you see it looks at low resolution, just like what we saw in tissue culture. Part of this aggregate sticks outside the cell. Part of it penetrates deep into the cell, almost to the nucleus, but it's all one particle, so it's all topologically outside the cell. And similarly, in vivo, we can see the actin polymerization around the areas where the cell is interacting with these aggregates of lipoprotein, which, as you can see, have become elongated as they interact with the cells. And this also was known for quite a long time, that most of the cholesterol in the atherosclerotic lesions is unesterified, free cholesterol. This was shown by the lab of Howard Kruth at the NIH around 1990. There wasn't any good explanation for it. And this was actually a pathology stain of philippin from a human aorta. And there were all these philippin deposits, which the pathologist observed, but really didn't know where they had come from. So there are some interesting consequences of this different way of macrophages taking up the cholesterol from aggregates of lipoprotein. So one is that if you treat this is again in cell culture, if you treat with HDL, you can remove the free cholesterol from the aggregates as it's getting made. And so you find less philippin labeling of the aggregates as you increase the HDL concentration. And similarly, less delivery to the cells. Interestingly, if you treat with the HDL, again, which is all extracellular in this case, you can significantly reduce the formation of lipid droplets inside the cell as seen by this lipid droplet stain lipid tox screen and quantified over here with various concentrations of HDL, which we used in cell culture. So what we think from this is that lysosomal acid lipase converts cholesterol esters to free cholesterol and to fatty acids outside the cell. The cholesterol remains associated with the aggregated LDL, leading to a very high concentration and high chemical potential of the cholesterol on the surface. And this, in fact, can lead to cholesterol crystal formation. And I should say, in regards to the HDL, it's been shown previously in vivo that HDL can accept cholesterol spontaneously from membranes with high levels of cholesterol, and this was shown by Mike Phillips and George Rothblatt a number of years ago. Now, this is a slide that I borrowed from a paper from the laboratory of Uerni and Kovanen. And what this shows, and you can read their interpretation of it here, is this is a large lipoprotein particle in a human carotid, and associated with this lipoprotein particle, they see this cholesterol crystal. And so this would be consistent with what we've been finding. And we also find some evidence for cholesterol crystal formation because the cholesterol in the aggregated LDL goes exceedingly high. And so it comes to the point where it just crystallizes out in solution. And in fact, in a human carotid, they find these cholesterol crystals associated with these large lipoprotein particles outside the cell. So I'm going to tell you a little bit about what we know about how this works inside the cell, what are the signal transduction pathways. And so the main thing that we've seen is that this is a process that requires the toll-like receptor 4 and its interaction with MyD8 and signaling pathways that are known to be associated with this, including the protein tyrosine kinase 6. And I'll show you the kind of experimental evidence we have for this. But first, well, as I say, it's been known that toll-like receptor 4 actually activates these various signal transduction pathways, leading to things we see like the actin polymerization. And it's been known for a number of years, 18 years, that if you knock out TLR4 or MyD88 in a mouse and look at the atherosclerotic lesions, in the heterozygotes you get less atherosclerosis, and in the homozygous knockouts you get much less. So this pathway has been known in some way to be involved in the generation of atherosclerotic lesions. So what we did was we took bone marrow-derived macrophages from, in this case, TLR4 knockout or MyD88 knockout mice, and we asked how much actin polymerization is there, as seen by phalloidin, associated with the aggregated LDL, as shown here. And if we do it in the TLR4 knockout or the MyD88 knockout, it's reduced by about 60 or 70% reproducibly. Similarly, if we look at the amount of lysosome secretion by the assay I described earlier, this is what you see with wild type, and you see deposition of the lysosomal contents onto the aggregated LDL, and you see very little of this in the TLR4 and MyD88 knockouts. And again, this is quantified over here. So we think that this is regulated by a signal transduction pathway, which is actually something that in principle one could use to intervene in this process. Unfortunately, all the molecules that we've found so far that are in the signaling pathway are widely used: PI3 kinase, AKT1 and 2, VAV, CDC42, and so on. We've not been able so far to figure out what are the RABs and the snares, which might be more specific molecules to understand what controls the lysosome secretion. But we're working now on developing this whole signal transduction pathway to really understand how this works in some detail. So the final thing that I wanted to talk about is how does cholesterol get from the plasma membrane to the endocytic recycling, to the ER, where it can be reesterified and form the lipid droplets that are characteristic of foam cells. Well, there's a pathway that I'm not going to talk about. It's been studied quite a bit recently with the astergram D1 proteins that are integral ER proteins, and they play a role in the transport at plasma membrane ER contact sites, and this is certainly an important thing to look at. We, for a number of years, have also been interested in these star D4 proteins, which were discovered by the laboratory of Jan Breslow at the Rockefeller University, and to play a role in non-vesicular sterol transport. These are small proteins that are soluble. They can easily move around in the cytoplasm, and we've shown that they can exchange cholesterol with various membranes. But actually, in a paper that was just published a few weeks ago, we found that they are especially good at taking cholesterol out of PI45P2 containing membranes, like the plasma membrane, and they're less fussy about where they transport the cholesterol to. And so this is actually work, our first studies of star D4 more than a decade ago by Bruno Mesmin, who is now at the University of Nice, Sophia Antipolis, but he was a postdoc in my lab at the time, and this was looking at U2OS cells. And first we were looking at what happens to the hydrolysis of SREBP2 if you don't have star D4 and you reduce cholesterol, and the answer is you get about a 30 or 40% reduction in the proteolysis of SREBP2, indicating that the ER is not sensing the reduction of cholesterol in the cells. And conversely, if you add cholesterol on a cyclodextrin carrier in control cells or in cells overexpressing star D4, you can get about a twofold increase in the amount of cholesterol esterification in the presence of star D4, indicating in both the sensing lower cholesterol or higher cholesterol, make changes in the plasma membrane, you can sense them in the ER, and if you reduce the star D4 concentration, you get a reduction in that sensing of the change in cholesterol. So we decided then to see what happens in our system with the aggregated LDL interacting with macrophages. And these are, again, bone marrow-derived macrophages from star D4 knockout mice that we got from the Breslow laboratory. And under wild-type conditions, you get a certain amount of cholesterol esterification, which you can see with the lipid topic screen when you incubate cells with aggregated LDL. And you get about a 50%, 60% reduction in that cholesterol ester lipid droplet formation in star D4 knockout cells. So it indicates that star D4 can take the plasma membrane cholesterol that comes from the aggregates, bring it to the ER, and allow it to be esterified. And there's no change, no significant change in the amount of lysosome exocytosis, indicating that the process is still going on. The cholesterol is getting delivered to the plasma membrane, but without star D4, it's not getting sensed back in the ER. And there's no change in formation of lipid droplets in star D4 knockout cells when you feed them acetylated LDL, which goes by the conventional pathway into late endosomes and lysosomes and leaves by NPC2, NPC1, et cetera. Okay, so so what we think then is that cholesterol in this unique situation of the atherosclerotic lesion enters macrophages directly into the plasma membrane. This activates certain signaling pathways, and other metabolites, including ceramide, are also generated and have effects on the cells. HDL or cyclodextrins can directly remove cholesterol from the aggregated LDL. And the free cholesterol gets to such high levels that it can actually form cholesterol crystals, which are known to be take part in inflammasome sensitization. And just to summarize, we think that this process we call digestive exophagy is used by phagocytes to degrade large objects, involves actin-dependent sealing, lysosome secretion, and VATPase acidification. This leads to extracellular hydrolysis of the aggregated LDL, and the free cholesterol in the aggregated LDL is the predominant form of atherosclerotic blood vessels. And the free cholesterol can crystallize, as I've said, and the cellular transport of cholesterol to organelles, including the ER, where it is esterified, and that can be mediated largely by STAR-D4. And so finally, I'd like to acknowledge the people who have contributed to this work. This really started when I was at Columbia in the 1980s, and I met Ira Tabus, who's a leading atherosclerosis researcher, and he and I started a collaboration that still goes on. And so this really started with him, and him making me aware of aggregated LDL in vessel walls, and all of the people, especially Abhi Heka, Raj Singh, John Ma, and Noah Steinfeld, and Frederick Lund have been involved in this work over the years. And I thank you very much for your attention.
Thank you very much, Fred. That's very impressive work. I may start with a question. So if the aggregate LDLs enter the lysosome and cholesterol reach the membrane, plasma membrane there, how come the NPC2 is taking care of the free cholesterol? It has to be released from the plasma membrane?
So there's two things that go on. So we think that it's actually direct insertion of cholesterol from the aggregate to this convoluted invagination. And so the cholesterol is delivered directly to the plasma membrane. Never reaches a late endosome and lysosome, never reaches NPC2, NPC1, et cetera. The lysosomal acid lipase works outside the cell to make the free cholesterol. Now, what you did see in the video I showed of the pH process is that some of the large aggregate gets broken off into what we call bite-sized pieces, and then it gets into a conventional late endosome and lysosome, and then you need NPC2 and NPC1 to move it out of the lysosome, potentially to get to the ER. But uniquely, in this situation with macrophages encountering aggregates of LDL, the cholesterol doesn't go to a lysosome. The lysosome comes to the cholesterol.
Thank you very much for your presentation. Many works demonstrate or show that smooth muscle cell became foam cells. And do you think that this is the same mechanisms then for macrophages?
That's the next thing I want to study. I don't think we know the answer to that. I actually read a review article that was written this month about how smooth muscle cells become foam cells. And it was still talking about endocytosis and all the usual things. But I do wonder if the smooth muscle cells encounter this lipoprotein particle that is so filled with high chemical potential free cholesterol, that if they touch it, the cholesterol is going to get transferred into their membranes, and then they will esterify it and so on. But somebody needs to show that. We will take it up some year, but if anybody else wants to do it, go ahead, because we've got plenty of other things to do. It's a great question. I wish, I've really been thinking about this for the past 6 months. If I had someone in the lab to do it, we would try to find out.
I have a question regarding the aggregates. What is the link between the HDL inside the aggregates?
Well, so the aggregates are cross-linked by ApoB, which is cross-linked by various proteins, including lipoprotein lipase, which is present to some degree, but it's known that it can bind to both ApoB and to extracellular matrix components. And so if you have several linkages mediated by, for example, lipoprotein lipase, you have this aggregate that is basically immobile and can't, because of its interactions with the extracellular matrix. Now, there may be other linkers as well, but that's the one that I know of that's biochemically characterized.
You showed that there is an activation of the signaling pathway inside. Do you think that this activation will increase the captation or the captation of the new aggregate or lysosomal moving to the aggregate?
Yeah, so that's, I think so. In other words, well, among other things, we see the TLR4 activation, and if we don't have TLR4, or if we block the various downstream signaling pathways, most of this doesn't happen. So you do need that intracellular signaling pathway in order for all of this to take place. We've also seen, we haven't published this yet, if we compare M1 macrophages to M2 macrophages, this process is predominantly seen in the M1 macrophages. And if you convert them to the M2 type, then you see very little of this process. So this is, we now think that this is predominantly a phenomenon of M1 activated macrophages.
You underline the role of actin polymerization and microtubules. Do you think there would be ways of action on the corresponding signaling pathways?
Well, in cell culture, but to do it in vivo, you would affect so many tissues and so many cell types. I don't think it's going to be a practical thing to do. I don't think I'm going to have time to take this to the end, but if I were, I can imagine two pathways to think about this right now for therapeutic implications. One is, there was a paper in one of the science journals about 3 years ago that in a mouse model of atherosclerosis, if you inject cyclodextrins, which are cholesterol chelators, you can actually reduce the atherosclerosis safely in the mouse models. And we know cyclodextrin can be injected into humans because kids with Niemann-Pick C disease are receiving this on a regular basis. The cyclodextrin will remove that excess free cholesterol from these aggregates in the lesions and take it elsewhere in the body where it can be disposed of perhaps more safely. So I'm a little bit surprised, and I've tried to get drug companies interested in this idea of following up, and I don't seem to be making any headway in this regard. But I think that's a possibility that could actually be acted upon now. And I think if we can figure out which are the RABs and the snares that are involved in this, that's where we're more likely to see something relatively cell and process specific that you could inhibit. There have been many studies of lysosome secretion and what are the proteins that regulate this in the cytoplasm. All of the known, we've tried everything that's published in the literature, and none of them are our pathway. So it's either a redundant pathway or it's a different RAB and snare pathway. If it's different, then that would be the kind of thing that I would look for therapeutic intervention.
Okay.
Yeah.
Thank you very much, Fred.
Merci.